Okay, the purpose of today's A-level A2 grade booster workshop is to revise on key concepts, or revise every chapter that you've covered for A2. Help students to understand command words in CAE examination. So you know that every question, they've got a command word to ask you what they want from you. So we want to know what are those command words and what do they mean, so we know what are the type answers that they are asking from the students. Help students to answer questions precisely and concisely in order to score the marks. Okay. So let's start with the first question. Choral, although they are, and so the first one is question which start with the keyword state. State. State means give a very concise answer with very little or no supporting arguments. So just one word, for example, a numerical answer that's easily be obtained by inspection. And state is very similar to name or identify. Okay, so question 1. Coral, although they are animals, are sometimes mistaken for members of the plant kingdom. So we thought that corals are plant, but actually corals are animals that are sessile. Sessile means they are attached to some solid base, so they will not be moving around, but they are actually animals. Okay, so question asks you to state 2 ways in which corals differ from plants. So that means you need to talk about corals. How corals differ from plants means you need to talk about corals, and they say corals are animals. That means they're asking you for features of animals versus that of plant. So this question, you have learned it when you learn the classification under the chapter 18, biodiversity, classification, and conservation. So always go back to the basic that you have learned. Okay. Where is that thing? The basic that doesn't look like it's here anymore. Okay. This is ecological sampling. So go back to the basic that you've learned. The simplest one is this particular table, which compare between the 5 kingdoms that you have learned. And the question asks you to compare between plant and animal. Talk about animal that's different from plant. So straight away from this table, you can see very clearly what are the differences, right? So number 1 is there is no cell wall. And number 2 is the way they obtain their organic materials is heterotrophic. So these are the 2 answers, right? The 2 answers they are given in the mark scheme also. Number 1 is there is no cell walls, right? No cell wall. And number 2, they are heterotrophic versus the plant, which is autotrophic. So you just have to talk about the animal because they ask for you. And 1 more mark is given for if a student say that, okay, there are no chloroplasts because the cell, animal cell, does not photosynthesize. So no chloroplast is also 1 mark. Okay. All right. So let's go to question number 2. Question 2, state the molecule that is required to react with ATP in order to convert ATP to ADP and inorganic phosphate. So in A2, you learn that all the breakdown processes so far that you learn in your A2 syllabus, they are all hydrolysis. Of course, there are other ways which are not hydrolysis, which you didn't learn. So therefore, in this case, when you break down ATP to ADP and PI, it is also hydrolysis, which means to say hydrolysis, you use a water to seal the wound after you lysis, means cut the molecule. So therefore, the answer is very easy, water. Okay, you can write water, the word, or H2O. Right? Okay, next question. Question 3. Name the membrane-bound enzyme. Do you think I need to make the question bigger so you can see? Yeah, okay. Name the membrane-bound enzyme responsible for phosphorylating ADP to make ATP. So the enzyme that I've taught you is ATP synthetase or synthase. So in this case here, remember, sorry, synthase is actually no H1, synthase. Now reject ATP, where is it? Set, no more. Reject, cancel, clear. Reject ATPase, because ATPase is a different enzyme. ATPase is an enzyme where it catalyzes this reaction, the hydrolysis of ATP, ATP plus H2O to become ADP plus PI. Inorganic phosphate means PO4, H3PO4. Okay, PO4, 3 negative, or H3PO4, PO4, 3 negative, or H3PO4. Okay, when all the O has a H attached to it, right? Okay, I think I don't want to go into more detail, which is AS stuff. Let's move on to the next question. Okay, if you want me to elaborate further, please unmute yourself or type in the chat so that I can actually go more detail towards that direction. Okay, number 4. So the figure shows some ways in which ATP may be synthesized and used in the cells. Right, so the question asks you to fill in the blank. Complete the figure 2.1 and state the correct terms or examples on the 3 dotted lines provided. So on the left-hand side, ATP is synthesized from ADP, so it's a synthesis. So you know that for cells, there are only 3 ways that ATP can be synthesized. All of them are phosphorylation, right? So you got oxidative phosphorylation, dash phosphorylation, photophosphorylation. So this dash phosphorylation is substrate level is the answer. Substrate level phosphorylation. Okay. Now, on the right side is ATP converted to ADP. That means ATP is used for certain processes in the cell. So anabolic reactions is a building up process from simple to complex molecule. So you name it, any processes that synthesize protein. So it's protein synthesis, DNA synthesis. DNA synthesis is also called DNA replication. Okay. Synthesis of glycogen. So it's glycogenesis. Right. Converting glucose to glycogen is also anabolic reactions. So all these are, they also accept polymerization. Okay. So these are anabolic reactions. Now, cellular work means when the cell do work. So you have got so many work that you've learned before, right? Like active transport, right? Muscle contraction, okay. Movement of vesicles on the microtubule. Remember the movement of organelles along microtubule? All of them need the ATP, sliding of the filaments. When you pull the sister chromatids apart during mitosis, there is a sliding of filaments, so you need ATP. Okay. Okay, and any other correct answers? Cellular work. Now, last one is, no, movement is nothing mentioned, cellular work. Cellular work, movement. Okay, so next one. So question 5. Glucose produced in the liver cells can be released into the blood to maintain blood glucose concentration. State one use of glucose within the liver cell. So this one has to do with your understanding of all the conversion of glucose to something else. When you start with glucose, what can you do with it? Okay, so you can, so it means pulling everything that you've learned about the glucose. So you may want to convert glucose to glycogen. You may want to convert glucose to CO2 and water in aerobic respiration. Okay, you may want to convert glucose to lipids. So the answer here is conversion of glucose to glycogen or lipid are all under the same category. Conversion of glucose, state one use of glucose is used for glycolysis, okay, or respiration or aerobic respiration, because anaerobic also can, aerobic respiration. So these are the two marking points that the examiners are looking for. Okay, so which means to say that you need to know very well that particular reactions. Okay, I'm showing you from your PowerPoint for respiration chapter. Let's go back to your respiration chapter. Substrates for respiration. Okay, let's look for substrate for respiration. Respiratory substrates, okay? All right. So it's actually from here, this one. You see, this is the pathway from protein, carbohydrate, lipids, the oxidation or breaking down of those food molecules into CO2 and water. So they all need different enzymes. Now we can also go, this is going down direction. The reactions can also be a going up directions in our cell, but going up directions will be an anabolic reaction. Coming down is a catabolic reaction. And both of them, both directions will use different enzymes. Okay, both directions, they use different enzymes. So when you want to synthesize lipids, okay, you can use any of the component here that link to lipids to synthesize them. All right. So that means you can use carbohydrate glucose to go through this pathway you see, glucose to go through this pathway and then synthesize lipids, glucose to go through this pathway and also synthesize lipids. It can also go through this pathway to synthesize amino acids and then protein, because they are all interlinked, but it's a different enzymes if you go upwards compared to going downwards. So therefore, once you know this, you know how to answer the question. Okay, and of course, glucose can also be used to synthesize glycogen. That is another chapter, right? It's another chapter, homeostasis chapter. So know your basic, then very easy to answer the question. Okay. So question 6, additional oxygen is required in the metabolic pathways involved in the conversion of lactate to glucose. So when you convert lactate to glucose, you need oxygen. State the term given to this additional oxygen. So you already learned from anaerobic respiration. Okay, let's go back to retrieve the information that they've learned. Where's my anaerobic respiration? Here. So in this topic, there's a graph that you've learned, which is this one here. All right. So y-axis is the oxygen uptake, right? Showing you this is the oxygen needed for basal metabolic rate. Basal metabolic reactions, it means when you're resting, whatever cells that still need oxygen to carry out respiration, this is the amount you need. Okay. Then at this time you start exercising, so you need more ATP. So you need more respiration, you need more oxygen to synthesize ATP you need. So this is the exact amount that you need. So at the beginning, when you cannot produce that exact amount, you have to go to anaerobic respiration to produce lactate. So these are the timing and the stages where lactate is being produced. You need to know this because I can give you this graph and ask you to point. So which region of this graph is where the anaerobic respiration is happening, or when the lactate is produced? It's actually this part here. Okay. So after you stop exercising, whatever lactate you produce in this stage of respiration, you need to convert the lactate back to pyruvate, and from to pyruvate. And then after that, for aerobic respiration. So therefore, when oxygen is sufficient, you're going to pay back the debt. So this part here is called oxygen debt. So this green color region. Whereas the blue color region is called oxygen deficit. You don't have enough oxygen, but you need that amount, so it's deficit. This is, I'm paying back now because I've got oxygen. So this oxygen debt, you can see this green color area is bigger than the blue color area. That's why they said there's additional oxygen that's needed. Okay. After you stop exercising, there's additional oxygen that's needed. Okay. That additional oxygen is the one above this region. Correct. So this entire green color region is the additional oxygen, minus this region here. Untrusted server certificate. Okay, trust. Okay. Now check carefully. They may ask you to shade the region. Remember, we do not consider this region here, which is oxygen used for maintaining basal metabolic rate. This region here is not considered, because this is the oxygen needed for basal metabolic rate. So only this green color region, highlighted region in green color, is the oxygen that's needed to convert the lactate back to the pyruvate. Okay, and it's actually slightly more. It's slightly more. That's because not only you use them to convert back to lactate, but don't forget, the moment you stop exercising, it needs some time for acclimatization of your muscles, such as your heart muscles is still pumping very fast, and the diaphragm and intercostal muscles, which are involved in breathing. Your breathing rate is still very fast. So even though you have stopped exercising, those two muscles, two sets of muscles, one is for the heart and one is for your lungs, your breathing, they are still working at a rate higher than the basal metabolic rate. So hence, the green color area showing the oxygen that is absorbed by the person after exercise is actually more than the blue color area, which is the oxygen deficit. So which means to say that the additional oxygen that's required for the working of the muscles of the heart, as well as the muscles involved in breathing, they are all the excess of the green color minus the blue color. So they can also ask you this kind of question. Okay. So that means to say whatever oxygen is needed to convert lactate back to pyruvate will be exactly the same as the blue color region. Okay. Okay, so let's answer the question. Back to the question. State the term given to this additional oxygen. So this is called the oxygen debt. Okay, that means you're already resting. How come you're still absorbing more oxygen that's required? More oxygen that's required from the resting body is the oxygen debt that need to convert lactate back to pyruvate. So 1 mark for that. So this question actually shows you how important it is for you to learn up the biological term, especially when this kind of question come out. It's very easy provided you have learned up the biological term. Okay. Question 7. Of the 125 cm³ of glomerular filtrate that enters the renal capsules each minute, only 45 cm³ reaches the loop of Henle. So that means when this 125 cm³ glomerular filtrate enters the renal capsules, it goes to PCT, right? Then after that, loop of Henle, isn't it? So that's what they're talking about it. So I've got 125 coming into my glomerular, coming into my renal capsule, and then go into the PCT. But only 45 cm³ reaches the loop of Henle. So when I come to loop of Henle, so here I've got 125 cm³. But when I come to loop of Henle, I only left with 45 cm³. So the question asks a student, name 2 substances that are reabsorbed into the blood from the PCT apart from water. That means to say that how come the volume has dropped? Okay, what are the things that has been selectively absorbed in this region? In this region, okay. Okay, so apart from water, so you know that there are a lot of mineral ions like sodium plus, okay, chloride ions, basically sodium ions. So sodium ions are being absorbed. So there's 1 mark for mentioning a named ion, or you write, if you cannot remember, right, you write something similar. They actually accept the word mineral ions. Okay, in this particular question, doesn't mean they will accept for all. So the best is still give the exact answer. Sodium ion, I guarantee you get 1 mark. Mineral ions, maybe certain year you get mark, certain year you don't get mark. Okay. Vitamins. So all the nutrients will be 100% reabsorbed. So remember, 80% of sodium ions are absorbed. 100% of the nutrients of vitamins. Glucose is a nutrient. Okay. 1 mark. Amino acid is actually 1 mark. Okay. And also we know that 70% of urea is being absorbed. So therefore, urea is also being absorbed, even though you want to remove it. Okay, but the nature is such that urea is being absorbed. So these are the answers they're looking for. No other answers. Okay. So this is what you've learned. So let's look at the charts that you've learned for this topic. So you are more familiar with it. Right, let's look at the A2 chapters. We're going to homeostasis, right? Kidney, ultrafiltration, PCT, PCT. So let's look at the components that being reabsorbed. So I've shown here very clearly. Can you see that? Sodium ion, 80%. All the nutrients, glucose, amino acids, vitamins, 100%. They actually didn't give mark for hormones. Water already mentioned, so no mark. Amino acids is given a mark. Protein, no mark. Urea is actually 40%. So 40% of urea reabsorbs. Urea has got mark. Okay. So next question. During stomata closure, state precisely where abscisic acid will bind. So you know that this abscisic acid is a plant growth regulator, a plant hormone called plant growth regulator, right? It is a plant growth regulator. So I always use this term. This is a biological term for plant hormone, okay? And this plant growth regulator, any hormone, you name it, whether it's in animal or in plant, it has to bind to a receptor on the membrane of the cell. So there can be a receptor on the membrane of the cell, right? A receptor attached to the membrane of the cell. So there could be a protein here. So they ask you for where abscisic acid will bind. So the answer here is very interesting. Some students may score, some students may not score, even though your answer could be correct, because there's certain keyword that they want. So Receptor alone, no mark, which is very interesting. State precisely where abscisic acid bind. For me, receptor is already correct, but for the mark scheme, receptor got no mark because receptor, this answer is actually bracket. My goodness. It's bracket. So receptors on the plasma or cell surface membrane, okay, of the gut cells is the answer they're looking for. That means they're actually looking for an answer where students say, okay, receptor, you know why is that there's no mark? Because there's a keyword here in the question, precisely, ABA. So that's the reason. That's the reason they don't accept receptor, because if you say, tell me where a PGR or a hormone binds, then you say, okay, it binds to receptor, general statement. But they say, tell me precisely where this ABA bind. So ABA is so specific. If you say it binds to receptor, any PGR like gibberellin, right, auxin, all of them bind to receptor. So it's no longer precise already because, okay, which part? So therefore the answer says receptors on the plasma membrane or cell surface membrane. I would want you to write of gut cells. That is called precisely. So that's why it is specific to that scenario that is given in the question. It's no longer a general question. So you cannot give a general answer where it does not apply, where it applies throughout regardless of it's ABA or not ABA. So I think that's the reason why receptor got no mark. Okay, so that's where you really need to write every single detail in your answer that is correct to ensure that you can score this 1 mark, which is a very precious mark for you to score A star or A. Okay, 1 more mark to go for A star is very precious. Okay, so you want to do that. Right, so question 9. Okay, so figure 4.3 shows the relationship between light-dependent and light-independent reactions in the chloroplast. So you're all very familiar with this, and very easy. So the question asks a student to name the substances X and Y in the figure. Very easy, right? Because I've taught you that in the light-dependent reaction, it will produces 2 molecules needed for light-independent reaction, which is Kelvin cycle. So ATP, because you need to convert light energy into a ATP, which is the energy currency of the cell. That means if you want the cell to produce anything, you need to pay any part of the cell using ATP. So you cannot pay using sunlight. So light-dependent reaction is to convert the sunlight to ATP. So that's why ATP is a product of light-dependent reaction. Okay, another thing is, of course, the reduced NADP is actually the answer. So that's the only answer they give. If you write NADPH2 or NADPH plus H plus, you will also get the mark. But you make sure you write NADPH2 or NADPH plus H plus. Okay, in words is reduced NADP. Okay, so they ask you to name the substances. So in words or in a formula form must be correct. Right. So far so good. Any question that you want to clarify? No? So let's move on to the next question. So D. muscipula grows naturally in a small region in eastern USA. Fires that destroy much of the vegetation occurs in this region. 3 sites in this region where there had been fires were identified and investigated. So site 1 has a forest fire 2 years before, site 2 is 10 years before, and site 3 is 30 years before. Preliminary observations suggested that over the course of time, following a fire, the biodiversity of plants had decreased. So that means after forest fire that wipe out the whole forest, when new plant starts to grow and animals come back in, it seems that the biodiversity of the plants had decreased. Sorry. The biodiversity of plants had increased, okay? Biodiversity has increased, interesting. However, the abundance of Venus flytrap plants had decreased. And so had the light intensity available to them has decreased. So which means to say that because the biodiversity of plant has increased, that means there are more different kind of plant species, right? You know, plants have got different level of height. So you've got plants that are very, very tall, okay? And you've got plants that are slightly shorter, right? And then you've got smaller plants, shorter plants. And then my Venus flytrap are very, 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 very short and stunted. So if you've got a lot of plant species, different kind of plant species, the Venus flytrap definitely cannot get enough sunlight. So that is a problem. So biodiversity of plant have increased, but then the abundance of Venus flytrap has dropped. So the total number of Venus flytrap per unit area. So abundance means the number of Venus flytrap or the percentage cover. Remember, when you measure abundance, remember what's a unit for abundance for populations? It can be the percentage coverage. That means out of total area, how much of the percent of the area is covered by this particular plant, or it can be total number of the plant per unit area of the area. Per unit area of the area. So per unit area. Okay. So you need to know the formula for abundance. So there was no, so let's continue with the passage. There was also a decrease in the nitrogen that the plants absorb from insects as a proportion of all the nitrogen they obtain from the environment. So there are less insects available for them to obtain the nitrogen. So available for Venus flytrap, okay? Name an ecological or statistical method to measure. Okay. First, the abundance of D. muscipula. Can you please underline these two words in your worksheet? Because it is a species name, so it must be italicized in the typing, and it italicized in the typing and underlined in handwriting. So it's not italicized here, so please underline it so that it's correct format. Now, they're asking you for abundance. So they ask you, what is the method, ecological method or statistical method to measure? So in this case, how do I measure abundance? Always go back to your syllabus. What have you learned for this method to measure abundance of a species? So let's go to your theory in measuring abundance. Oh my God. There's too much information here for me to retrieve. Let me see. Abundance is this one, I think. Or this Pearson's correlation. Okay. Measuring abundance. Okay, here. Measuring abundance. Yeah. So you have in your syllabus, you have learned only 3 ways of measuring abundance of a species. These are the 3 ways, right? Either random sampling using quadrat, or systematic sampling using line and belt transects, or capture mark recapture. So only these 3 methods. Therefore, the answer that you need to write must be either of these 3 here. Okay, so let's go back to the question. You want to measure the Venus flytrap, right? So of course we cannot use capture recapture technique. We will also not use the line transect because there is no clear gradient along the line. There's no clear gradient along the line, so it's not necessary to use line transect. So therefore, the only method for this answer is, of course, random sampling. Okay, go back to this one. Random sampling using frame quadrats. So you need to tell me, are you using frame quadrats or point quadrats? Because quadrats got two types, frame or point. So the answer for this one is you need to use, where is the answer? Here, go back to the question. So the answer is random sampling using frame quadrats. Please make sure the spelling of a quadrat is correct. A quadrat, there's no E behind there one, okay? So your spelling must be correct, because I saw the mark scheme in paper 5 that spelling must be correct to score the 1 mark. So please learn up the correct spelling, okay? And learn up whatever that is in your syllabus. So this is the answer. What about the ecological or statistical method to measure plant biodiversity? So you know that you have learned Simpson's index of diversity. So the answer is Simpson's index of diversity. Third, the strength of relationship between light intensity and proportion of nitrogen absorbed from insects after taking measurements from a large number of plants. So you know that this is actually asking you for correlation. So you have learned two types of correlation. First is the Pearson's linear correlation. Let me write down first. Pearson's linear correlation coefficient. So this is the first one you've learned. And the second one that you have learned is actually Spearman's rank correlation coefficient. So between these two, right, only one of them is correct. What do you think, which one is the correct answer? Alicia Ang? Which one do you think is the correct answer?
I wrote Spearman rank correlation.
Oh, why?
Because it has a normal distribution. I said.
How do you know it's a normal distribution? That the question didn't state that, but this is the correct answer actually. Any other reason other than this?
I'm not sure.
Okay, Pearson's correlation is when the relationship between the two variables here, which is light intensity and proportion of nitrogen, must be linear, right? Correct? Hello?
Yes.
It must be linear, right? That means to say that it's either like this, any version of this, right? Or any version of this. Correct? Any version of this. But for Spearman's rank correlation, it can be any version of this or this, right? Or any version of this or this. So which one gives you more choices? Sorry, it can be drawn here. Pearson or Spearman? Spearman gives you more choices, right? The relationship can be a linear or non-linear. So because of that, the only answer they accept here is Spearman's rank correlation, because there's no information given to us that is linear. So therefore, the safest way to do the correlation test is Spearman, because Spearman applies throughout, be it linear or non-linear. But it must be monotonic. Remember, monotonic. I hope it's the correct term I've introduced. That means as X value increases, Y value will also increase. This is called monotonic. Okay, so that's why the answer is number 2. So number 1, no mark one. So that means to say you really have to think of all the possible choices that you have learned in your syllabus. Which is the best one? Which is the most precise or accurate one? You only give that one. Okay, you only give that one because you want to score full marks for every question. Remember, every question you want to score full marks. And then you will not be surprised that all of you can score A star in exam 1. All of you can score A star 1 because I've given you the secret of scoring A star. Okay, so the secret is you have to study. You have to know, but what is it exactly you need to know? Okay, everything that's in the syllabus. So as long as you got time, you go and study all of them. You can see that every day you study a bit, right? You are adding more marks to your answer already, right? It's still a long way to go to the exam. So this is an advice for me. Trial exam is in 1 week's time, right? Is it? Yes. Trial exam is in 1 week's time. Now, if you have not started to do your revision properly, then please don't rush and try to revise all the chapter this week. It defeats the purpose. Why not you check from today until the actual exam, your A-level exam, how many more weeks do you have? So for all these weeks that you have, you distribute evenly the chapters across the weeks. So let's say you got 6 chapters and you have got maybe 12 weeks to go. Then every 2 weeks, you try to cover 1 chapter. So therefore, every 1 hour when you sit down, you try to cover a little bit, a small chunk, small chunk or small bite size from your syllabus, you will remember better. Okay, so that's how you approach studying biology. So you really have to start early and revise early. And then as you revise, try to draw summary chart or diagram or mind map. Because that is all what you understand. If you put in words, it actually enhances your memory and understanding of that particular topic. Okay, so that's very important. So if you do not need the trial exam results for you to apply to uni, don't focus on trying to revise everything for your A2 exam. Okay, you can write in your exam paper, maybe top right-hand corner, teacher, I have only studied this chapter for this exam. So I will know like, okay, for this chapter you've studied, you are able to answer the question, and for those chapters you're not studying, you're unable to answer the question. It's okay. But it's okay because it's showing you where you have covered. All right, so don't worry about trying to study everything for the trial exam. You are focusing on scoring as high as possible for your final in a way that is comfortable for you. Don't rush, don't stress yourself out. It's not necessary. Okay. Question 11. I hope that advice helped you with your strategy in revision. Okay. If you need more help on that, please let me know. I can actually talk to you personally and see how we can go about planning your revision, especially some of you who say they're sinking. Okay, please come to me. I'll help you to float. Okay, come to me. I'll help you. Okay, don't sink quietly and then after that you're under the sea. Really, I also don't know you're under the sea. I'll give you some oxygen tank. Okay, or I'll give you the float to float. Right, there always are ways, but you have to get help from people. Right, there are a lot of people who can able to help you, but you have to let them know. Okay. Question 11. Most reflex arcs pass through the spinal cord and involve different types of neurons. Name and state. So 2 keywords here, name, then state. The functions of the 3 types of neuron in spinal reflection. So this is very straightforward, right? So what are 3 types of neurons you have learned and what are their functions? So it's so straightforward. So let me pull out I'm pulling out too many PowerPoints. Shall I pull out the PowerPoints? Okay, pull out PowerPoints for you to see. So this is a homeostasis chapter, right? No, it's control and coordination, right? Okay. Right, so it is the reflex arc topic. So where is the answer here? Here, okay. So the answers here is, you see, name the neuron. So you got sensory neuron, interneuron, which is also called relay neuron, and motor neuron. So these 3, and because it's 3 marks, you must answer in pairs, means name and function correct, 1 mark. Name and function correct, 1 mark. So function of sensory neuron is to conduct impulses. Now, in the mark scheme, right, they didn't specify that student must write electrical impulse. They write their impulse only. So that means if you write impulse, also you can get marks. But to be more safe, I would highly advise you to write electrical impulse. Okay. Because I remember seeing past year question that if the student write impulse, you will not get mark unless you write electrical impulse. So to be safe side, please write in electrical impulse. So conduct electrical impulse from the receptor. So from receptor is actually 1 mark. Okay. To spinal cord or CNS, they didn't actually give mark because over here in interneuron, if you say is to conduct impulses from the sensory neuron to the motor neuron is 1 mark. And the third one is motor neuron will conduct impulses from spinal cord or CNS to effector. So you get all the full marks. Okay, so this is very easy and very direct. Right. Next question. Oh, okay, so question 12. State how the RQ value is calculated. So let's check whether you can score full marks or not, even though this is a very straightforward question. First marking point is given for if you say that it is actually the component is CO2 divided by O2. All right, this is CO2, actually, sorry, produced. You have to write CO2 produced versus oxygen consumed or absorbed. So this is first marking point. So please remember to write produce and consume. And then either number of moles or volume. So if you forgot to write moles and volume, you will not get the full marks. Okay. And of course, I also taught you that during respiration, but they didn't actually give mark for respiration. No mark is given for this question. So that means the more important 2 points are CO2 produced and moles or volume of CO2 and oxygen. Respiration is in bracket, means no mark. I think why respiration got no marks because this particular question is a partial sub-question in the main question, and maybe in the passage in the main question, they already said respiration. So because of that, no mark given for respiration. So I think there's a reason why this is not a marking point. So usually they will not give a mark when the passage of question already got that particular keyword. So you copy from the question and try to get mark. Of course, you cannot get lah. I think that's the reason why. Okay. Okay, next section is what is the keyword of the question is define. Okay, define. Define is a formal statement. Define is usually definition. That means it's a formal statement. That means your answer must be concise and precise like dictionary or a paraphrase. Okay, paraphrase. So let's define the word bioinformatics. So what are the words? What is the definition of bioinformatics that you've learned in your syllabus? Okay, let me look for it. Where's my bioinformatics? Is it this one? Yes, it's this one. Okay. Now, when you learn bioinformatics, right, this is in your PowerPoint notes. It's a biological study that involves the collection, processing, and analysis of a large amount of biological information and data, biological data, using computer software. Okay, so therefore the marking point, the marks given are for computer or software. Computer or software is 1 marking point. Okay, you write computer software is 1 mark. Computer is 1 mark, software is 1 mark. So it involves computer, it involves software. It involves the analysis of, so the word analysis is there, of biological information or data. So analysis of biological information or data is actually 1 mark. Okay, and 1 more mark given here is actually databases. Databases. Because when we learn this topic, right, we say that there are a few official database where we can retrieve the information, the biological information and data. So because you have learned this word database, database is also one of the point to define bioinformatics. And this, it is part of this field called bioinformatics. So these are the only 3 marking point for definition of bioinformatics. Okay. So which means to say, please memorize the whole sentence for a definition of a biological term. So the moment you memorize the whole sentence, you get the full marks. Okay. This is the secret to scoring A star in exam. Memorize all the biological terms. When you memorize all biological terms, it's a secret to scoring A star. Okay. And the second secret is do the past year question. Okay, that's the second secret. Do the past year question. Okay, question 2. Define the terms dominant and allele. So this again, if you memorize the definition, I guarantee you score full marks. Okay, so we go back to your notes where these 2 words appear. These 2 terms appear, okay? So let's go to the PowerPoint and check your definition from there, right here. So number 1 is allele and number 2 is, what is the definition? Okay, dominant and allele. So let's look for the definition. Dominant. Dominant here, see, this is dominant. You don't want to shift my things. Dominant. So it is the allele. So when you say dominant, you're referring to the allele from one parent that influences the appearance of the phenotype, even in the presence of an alternative allele. So the marking point given here, if you write this whole sentence, you will get the mark. Okay? Or any other alternative explanation which have the same meaning to this sentence. So what else can you write as similar to this sentence score other than this sentence is you need to say that it is an allele. So without the word allele, you will not get the mark already because we are referring to the allele. So an allele that is expressed in heterozygotes. So that's an alternative answer. Allele that's expressed in heterozygote is similar as the definition here. The allele that will influence the appearance of the phenotype even in the presence of an alternative allele. So it's actually heterozygotes. Influence the appearance of phenotype means it is being expressed. Okay, so this is an alternative word for the same meaning. So that's the only alternative that can score. Okay, the next definition is allele. So allele is the alternative form of a gene. So where's my definition for allele here? Alternative form of a gene. So you write alternative form of a gene, you score the marks. I don't want to give you an alternative answer. Just memorize this one, okay? Simple, straightforward, score full marks is most important. Okay? Yeah, no need to learn so much things. Learn the most important thing. Okay? This is strategy. Okay, the next keyword in the exam question is list. List means you write 1, 2, 3, 4, 5, right? So list is same as state or name, but it requires a multiple answer, means a few points instead of state. It means 1, name means 1, list means a few. Okay, sometimes they'll tell you exactly how many you should write. Okay, so if a specific number of points is requested, make sure you only give the number of points here. Now this one, okay, how is the mark scheme given? Usually it's like this. Now if the question said list 2 ways and they give you blanks for you to fill in, okay, that means you can write 3 or 4 or 5 and they only mark 2 correct answers. Okay, that means you can write as many as you want as long as you got 2 correct. But if they write there 1 and then you got the spacing for you to answer the question, and they write 2 and then you got spacing for you to write answers, if you go and add yourself, I'm not sure whether number 2 is correct. Why not I add 1 more so that I get the full marks, 2 marks. Sorry lah, if you add like this, right? This is how they're going to mark. Okay, this is correct. This is correct. And this is wrong. It's wrong. So how many marks they'll give you? They calculate this way. Full marks is 2 marks. Mistake is 1. Total 1 mark. So that's how they give you marks. So therefore, don't go and add additional points if they write there 1 and 2. Unless they didn't write, then you can give more if you're not sure. So therefore, give the one that you're sure. Okay? And don't add some more, because add some more also no use, because add some more wrong also minus. Can you imagine you add some more, right? 1 is correct, 2 and 3 is wrong, 4 is correct. You get 0 mark, right? Because 4 answer you give, 2 answer correct, and 2 answer wrong, right? So that means 2 minus 2 is 0. So that means 0 mark. So you better choose the 2 that you're quite sure and write there. Then you get the 2 marks out instead of 0 mark. Okay. Okay, question 2. Hey, did I answer the question just now? Oh no, I have not answered the question. Right. List 2 ways by which water may be lost from the body. This one is like AS answer, AS question. How water can be lost from the body of the mammals? So any ways where water come out from the body, you name it. Urine can, right? Sweat can or not? Sweating can, right? What about tears? What about saliva? What about feces? Feces also got, right? Because feces is semi-solid, means got water. Okay, it's not solid. So got water come out from there. All right, I think these are the answers. No, I think these are the answers. Any more answers? Yes, got bleeding. Right, you injure yourself and then it bleeds. So bleeding also. You go for operation, you bleed. Okay, same thing. So these are all the answers. And one more thing. Water vapor, no? Water vapor from where? From exhaled air. This is a very good answer. You know, the air that we breathe in could be drier than the air that we breathe out. So that means to say that a lot of water that evaporates from our airways, the surface of our airways, which is trachea, bronchus, bronchiole, and alveoli. So that's why the air that we breathe out is more humid. That means water can be lost from there. So this is a very good answer actually that we may not think of. So question 2. So before I proceed, do you have any questions or not? All of you? You're okay, Right, question 2. Some animals do not breed well in captivity. Oh, so it's like, remember zoo? In zoo, there are 3 rows, right? Right, we'll go to that later on. So list the methods of assisted reproduction that may be used with animals that do not breed well in captivity. So there are 2 things we want to revise here. Number 1, what is the role of the zoo? Number 2, what are all the methods of assisted reproduction? Okay, so let's go to the notes, PowerPoint notes. Where's my animal? Where's my assisted reproduction? PCT, no need. So no need. Here. First, I want to know the role of zoo first, okay? Role of zoo. Okay, so zoo has got 3 roles, same as botanical garden. Number 1, captive breeding. Botanical garden cannot use the word captive breeding, because you don't have to go and capture the plant because plant don't move. Okay. So zoo captive breeding. Number 2, after captive breeding, when you increase the numbers, number 2 is you want to release the captive bred individuals to the wild or to the nature. Then of course, the third one is to do research. Research. So that is for function of the zoo. Just now we talked about plant, right? Botanic gardens. So what is the role of botanic garden? So instead of captive breeding, the role of botanic garden is to protect and propagate. So the word is propagate endangered species of plant instead of captive breeding. So one is for animal, the other one is for plant. So you use different word for different organism. Okay. So what are the ART? Please remember the word ART. ART stands for assisted see here, assisted reproduction technology. Technology, this word means a procedure, methods. Technology stands for procedure or methods. So assisted reproduction methods, you see here, methods, okay? They are all summarized in this PowerPoint. Look for the words in red color. The words in red color are the mark scheme answer. Only these 4 words, nothing else. Okay, nothing else. Either you write these 4 words or you describe these 4 words. What these 4 words stands for. So you score full marks. So what is ART? Number 1 is AI. Okay, artificial insemination. Right, artificial insemination, or you describe what is artificial insemination means you inject the semen to the uterus of the female. Okay, or number 2 is IVF, in vitro fertilization. Right, for in vitro fertilization means you need to collect the sperm and also collect the egg from the organism. And you do the fertilization means you put the eggs and sperm together to fertilize outside the body. In vitro means in a test tube or in a glassware. So this is done outside the body. Okay. Whereas artificial insemination is actually done inside the body because you introduce the semen into the uterus of the female organism. So that's why it's actually done inside the body, whereas IVF is fertilization is outside the body. Okay. Then after that, the third is embryo transfer. So embryo transfer is usually whether you do AI or IVF, you can do embryo transfer. Because if you do AI, that means you let the fertilization happen in the uterus of the female organism of the cow. The next step is you might flush out the embryo to collect the embryo. So it's same as IVF. When you got after fertilization, you got a lot of embryos. So you will want to transfer the embryo to another cow, which is not the original cow where you get the ovum from. It could be a surrogate mother or foster mother. So embryo transfer is 1 mark. Okay, and then when you transfer to the organism, it could be a surrogate mother. So surrogacy is also a type of ART because maybe the captive bred organism is endangered. So you don't want to overstress the organism to carry the fetus to go through the pregnancy. So you choose another organism that is closely related. It could be a different species, but it's still mammals, closely related to this organism, and use the uterus of this surrogate mother to grow the embryo and the fetus. Okay, so these are the 4 points in the mark scheme. So I have updated the PowerPoint notes so that it contains all these 4 red color points today. So if you want the latest version, you can actually download from the PowerPoint which I've updated today. Okay. Okay, let's go to the next question. The next question is calculate. The keyword of the question is calculate. Now you need to remember that when you're asked to do calculation, working should be shown. Especially when you do working means you must show the formula and the numbers, slot in the numbers correctly is 1 mark. And after that, you calculate and get the correct answer with the correct number of significant figures, and the unit is actually 1 mark. So the mark is given for formula as a correct and the calculation, the working, and secondly, correct answer with correct number of significant figures and units. So these 2 points will be 2 marks. Okay. So let's look at the calculation questions here. So they ask you to calculate the percentage, right? So this is a very straightforward answer. They even ask you to give your answer in whole number. So it's very, very direct. So we don't have to calculate it. Okay, we know how to calculate it. Next, over here, they ask you to calculate and then you give 1 decimal place. So again, this question is very direct. I don't want to do the calculation unless you request me to calculate because it's very direct. It's actually 125 divided by 700, okay, times 100. So that will give you the percentage. So give your answer in it is equals to 17.9%. So working 1 mark, correct answer with 1 decimal place is 1 mark. Right, so next is determine. A question with start with the keyword determine means you need to determine the quantitative answer using the information given in the question. Or you're required to carry out some procedure to find out a numerical answer. Okay, determine means go to the passage to find out the answer from the passage or try to work out the answer from the information given. So let's look at sample question. Cloves are aromatic flower buds. So if you want to know what is clove, I have actually, where is my, okay, this is, let me show you what is clove. I think there. So this is clove. This is the, what do you call it? Herbs, spices that we use for cooking, clove. So cloves are aromatic flower buds of the tree eugenol. Eugenol is an alcohol. All is always alcohol. So eugenol is an alcohol. It's a drug extracted from cloves. Eugenol affects the movement of sodium ions through the cell surface membrane of sensory neurons. So you know that whatever neurons that we have, in order to maintain the resting potential and to produce action potential, sodium ions have to pass through the membrane, right, in different directions. So this graph shows you the effect of eugenol concentration. So the effect of eugenol concentration on the sodium ion movement. So the higher the concentration of eugenol, it decreases the percentage of sodium ion that can pass through the membrane. That means it inhibits sodium ion from passing through the membrane of the neurons. Okay. So use figure 6.1 to determine the percentage decrease in sodium ion movement at eugenol concentration of 0.5. So this is a very direct question. So they ask you to go to find out from the graph at a 0.5 concentration of eugenol, how many percent of sodium ions will be inhibited from passing through the membrane? So the answer from here is, you need to read accurately, is 58. So the answer given in the mark scheme is 57 to 58% only, because actually if you read correctly, right, it's slightly lower. So it's somewhere here actually, slightly lower. So that's why they also accept answer for 57. Okay. Any graph must be read accurately, right? So the next one is sketch, means ask you to draw. Very interesting question. They ask you to draw. So sketch means a question with the keyword sketch means either you need to sketch something on the graph given. So you overall outline the shape. Okay, the shape must be correct, or the position of the curve must be correct. Okay. So they require your freehand drawing only. Should take care over proportions and show important details clearly. With sometimes you may want to label an annotation. Sometimes they'll ask you to sketch and label. All right, so let's try out the question. Sketch usually use your freehand to draw the shape which is aligned with the theory that you've learned for that particular topic. So let's try out this question 1. The length of sea harangus is a type of fish, shows a wide variation. So the length of this fish shows a wide variation. So figure 8.2 shows a number of fish of different length in a population of sea harangus. The arrow shows the selection pressure P and S. Okay, let's look at the diagram here. So the y-axis is the number of fish, and the x-axis is the length of the fish in cm. So that means the x-axis is the range of the variation for the phenotypic variation of this population of fish here, and this one here, which is the peak, right? When majority, you've got the highest number of organisms having this phenotype, is called the optimum phenotype. Okay, so the question asks the student to sketch a graph on the axis below to show the distribution of the length of the fish when selection pressures P and S operate for a few years. So selection pressure is an environmental condition that will affect the survival of the organism, right? Environmental condition that affect the survival of organism. And usually when you say pressure means it will actually, when you're pressurized, do you survive better or not? Usually you don't survive that well. Which means to say, when the environment selection pressure act on this particular phenotype, so they will not survive very well. That means they're selected against survival. So when this act on this phenotype, it will not survive so well. So what will happen now is, whichever selection pressure act on the phenotype is selected against survival. So your drawing for the new distribution curve for the phenotypic variation of this population should be, cannot draw why, should be, cannot draw, can draw already, okay, should be, I don't know why they don't allow me to draw. I cannot draw on the graph, I can draw at the side. Why? I cannot draw on the graph. Okay, never mind. I try again. Can draw on the graph already. Okay, try again. It's 30, sorry, it's here, right? So this is the point where it's selected for survival. This phenotype is selected against survival. Organism with this phenotype is selected. So it's, I cannot draw again. Why? I can already. Okay. The left side and right side should be symmetrical. Can I draw? Okay, and then symmetrical. Assume that left and right symmetrical. So that's the answer here. Keyword for the answer. The optimum phenotype is still the same. So that's 1 mark for optimum phenotype located in the same place as the initial optimum phenotype. This is final optimum phenotype are the same. And then your distribution of the curve is narrower. And how narrow is narrow? It must actually stretch from number 5 here to number 35 and nothing else. Because this 5 and number 5, right? The length at 5 and number 35 are already selected against survival by the selection pressure. So it should not be there in this population anymore. So that's why you cannot draw there's no value here. It should be 0. Okay, so within this category. Right, part 2. Sketch a graph on the axis below to show the distribution of the length of sea harangus when selection pressure S alone operates for a few years. So S is on the right side, right? So that means it will lead to directional selection, where the extreme phenotype is located here. This area here, right, is considered as the extreme phenotype. So it's selected against survival by this selection pressure. And this is called all this phenotype here, this range of phenotype here is called the extreme phenotype. This selected against survival, extreme phenotype. So after some time, they are selected against survival and they're no longer there. So your new curve must not have, from 35 onwards, must not be there, but it's shifted towards the right side. So therefore, I will draw something like, this. You don't allow me to draw. To try a few times. Okay, so skewed to the right side. Okay, a curve that's skewed to the right side, where your range is from 0 to 35. Your range can be anywhere from 0 to 35 only. Okay. So can you see that they can ask you to sketch any graph? So in your syllabus, in your topic, any particular topic that has got graph, please pay attention. Right? For example, reabsorption of sodium, ion, glucose, potassium, urea in the kidney. So that one, they may ask you to sketch the graph. So you want to be familiar with those things. Like just now, the graph I show you, oxygen consumption by the body at different stages of exercise. So before, during, and after. So you also need to know how to sketch the graph for that kind of graph. So all the graph, please make sure you know the theory answer. Please learn all of them. Okay. Okay, shall we try one last question? Oh, finally we come to explain question. Okay. Explain is they're asking for long answer. The keyword is why. Why this thing is happening. So give reason. Give reason or reasons to your answer. Okay. That's what they're looking for explain. So let's try out the first question. Explain how the structure of glomerular capillaries is adapted for ultrafiltration. So they're only asking you for capillaries, but it's a 2 mark. So what are the keywords to write to score the 2 marks? Why not we retrieve the PowerPoint note so that you have got a better impression of the answer. So this is a homeostasis question. Okay. Homeostasis question. Ultrafiltration. So when we will learn about the structural adaptation for ultrafiltration. Right, this one here. Structural adaptation for ultrafiltration, there are 3 main layers that are involved in the ultrafiltration. That means the blood plasma must pass through 3 layers, right? The first layer is actually this layer here, right? This first layer here. So the first layer is actually the pores on the endothelium of the glomerulus. Okay, the pores on the endothelium of the glomerulus. So this is actually the answer for this question. Because there are pores, or another word is fenestration. Fenestration means having pores. Okay, this is a word here. Fenestration means having pores. Okay. They also accept gaps. They also accept holes. Okay. Fenestration or pores or gaps or holes on the capillary endothelium is a key word. Without the word endothelium, you will not get a mark even though you say pores or gaps, because there are 3 layers. Which layer are you talking about? Okay. And then, but this is not the main filtration barrier. The main filtration barrier is the one in the center, the middle layer, which is actually this layer here. Why is it blue color? I want to change this layer here, okay, the red color one. So this is the main filtration barrier because the spacing between the protein fibers are very narrow. So only molecules which can pass through this spacing can pass through the 3 layers and become glomerular filtrate. So this is the main filtration barrier. This is actually the basement membrane right here. So basement membrane is the main filtration barrier. Keyword basement membrane is 1 mark. So this is a second marking point. Okay. And then what is the size of the molecules which can pass through is any molecules where their size must be less than 68,000 relative molecular mass. Okay, or relative formula mass. So this is actually 1 mark. So any substances less than this one can pass through. Any substances or molecules more than this size cannot pass through. So please memorize this number plus the unit. Okay. 1 more mark is given for no cells can pass through. Okay. So what are the things can pass through? What are the things that cannot pass through? So these are the marking point. They didn't mention about the third layer. Okay, the third layer here is the gaps between the podocytes, right? The gaps between the podocytes is actually the third layer, right? So they don't give mark for the third layer because it's very big. So this is not the main filtration barrier. So let's read the question again. Explain how the structure of glomerular capillaries is adapted for ultrafiltration. I know why the third marking point, the third layers are not mentioned, because the question asks student for glomerular capillary only. So glomerular capillary, the capillary wall is made up of the endothelial cell, and the endothelial cell is actually squamous. Remember squamous? And all endothelial cell and epithelial cell of the body actually rest on another layer, the gel-like layer, which is called basement membrane. So that is why the mark scheme for this question only give mark if you mention about the pore of the cell, between the cell, as well as if you mention about the basement membrane, because this is part of the endothelium layer. So that's why if you talk about the filtration slit, actually the keyword is called filtration slit. If you talk about filtration slit of the podocytes of the renal capsule or Bowman's capsule, you will not get marks because of the nature of the question. Okay, wow, 3. So I think I will stop here today. Thank you for being here, and before you leave, please give me a feedback on today's revision workshop. Let me know whether number 1, the pace is okay. Okay, number 1 is the pace. Number 2 is whether it is helpful in your revision. Okay. And also number 3, there are three things I want you to let me know. Number 3 is whether you want me to continue to discuss the remainder question for next week. So I need 3 feedback from you. Number 1 is, I also forgot what's number 1. Number 1 is what? The pace, okay? The pace is okay, not number 2, helpful, not number 3. Do you want me to discuss next week? So can you type there number 1, 2, 3, and please give me your feedback so that I will know what to do, whether this is okay for everyone, whether it's helpful for you. So can all of you give me your feedback, please? There are 30 of you here, so I expect 30, if possible, that's the best. Please give me your feedback. I'll really appreciate that. Have I typed for you already? Okay. So please give me your feedback. Now, teacher, can you send the Maxim to us? No, I cannot scan. I will not send the Maxim to you. You need to attend the workshop and we'll discuss through and then you write down the answers. Okay. I will also ask Miss Annie, who is also conducting the workshop, whether she sent to her students or not. Okay, if she doesn't send, I will not send her. Okay, we need to standardize what we do. Okay. All right. So please continue next week. Choi V, okay, thank you for your feedback. Sophia, number 1. Can all of you type a feedback, right? So that I will know that I really appreciate that. For me, I find the pace is very, very slow, but I cannot go that fast because at this pace, I think you will be able to process what I share with you and you're able to understand. And at that time, you already understand and remember them already. I think that's why I go very slow today, very slow. Quality more important than quantity, I think. Right, we'll continue next week. Okay, any more feedback? Jaya Ganesh, thank you very much for your feedback. Yeah, I really appreciate that. No more feedback? Okay, if no more feedback, then thank you for being here. I will see you next week according to the normal weekly lesson. Okay, normal weekly lesson means if that day you don't have, it's online, means online, online recording. If that day you have, then we will have just like all the previous weeks, okay? So I will be recording as well if on that day is your tutorial and we don't have a live tutorial, I'll be recording with another class. All right, workshop's great. Thank you, Faith. Thank you so much. Faith always give feedback. Okay. Thank you for the workshop. It was great. Yeah, thank you. All right, so that's all for today. And I wish you a happy weekend and wish that you can achieve a lot for this weekend in your revision. All right, and I'll see you next week. All right, bye-bye, everyone. Okay.